Welcome, everyone. My name is Renee Duprell, and I'm the Director of Development here at ISB. Thank you for joining us. This is our first research roundtable of 2022. This is our virtual series that we host almost every month, and it's to keep you updated on our research. And this is also meant to be for the community. You do not need a PhD to join us today and keep up with us. Our intention is to have one of these just about every month. And we will keep you updated on that. You should hopefully be on our email list, so you'll see those coming, and I'll talk about our upcoming events after this. Today we'll be hearing from our featured scientist who's sitting right next to me, and then we'll go into a Q&A session. And you are welcome to submit questions at any time while Jim is talking. Please use that Q&A button at the bottom of your screen. And when he's done with that, we'll move through those methodically. So our speaker today is our own president of ISB, Jim Heath. He has directed the National Cancer Institute funded NSB Cancer Center since 2005. He was named one of the top 7 innovators in the world by Forbes magazine. So Jim, I'll let you take it away.
Thank you.
And you want to share a screen?
Yeah, I do want to share a screen. And I'm sharing.
Is that?
And one more. Oh, we just did this.
Is this
right?
it?
That's it.
There it is. Okay.
Okay. So first, welcome. I'm delighted to have a chance to talk to you all today. What I thought I would spend time on, and I spent the last couple of years studying COVID and more recently long COVID, and I'm happy we have a very large article coming out in the journal Cell sometime this week. I'm happy to answer questions about that. But I thought I would really talk about the Omicron variant and what we know about it. And so I'm going to walk through, I've got maybe a dozen slides on, for anybody who needs it, a refresher on exactly from the sort of virology point of view, what the coronavirus does, how it infects cells, and then how that is changed by the Omicron variant. And I'll say at the outset that, in general, what you would expect a virus to do as it goes through mutations is to become more infectious because it spreads easier, and less virulent because the carriers who get infected survive and continue. So basically, is if you just think about some sort of Darwinian selection process to propagate the virus, those two things are sort of part and parcel what happens. And what we've seen over the past year and a half or so up through the Delta variant is that the virus got more infectious, but it didn't necessarily get less deadly. And it seems like we're now moving into a phase where Omicron for sure is a lot more infectious, but it does seem to be less virulent. It still is hurting a lot of people, but I'm hoping that we're on a pathway towards a pandemic that we can live our normal lives with. And I'll walk you through that. So I'm sure everybody knows what this is, but this is a reconstruction of the coronavirus, the intact coronavirus, based upon a cryo-electron microscopy. And these green sticks that are coming out, those are the famous spike protein. And I'm going to talk quite a bit about what that spike protein is, because that really is the only thing you need to think deeply about in terms of vaccines. It is also where the major changes are taking place that is influencing how Delta and Omicron are different from each other. Okay, so this is the spike protein. This is one of those green stalks from this picture here. And when this was first, structure was first resolved, it became kind of apparent why the immune system somehow misses coronavirus initially. And a lot of that's because it has these, if you look at these gold groups on here, these yellow gold groups, those are sugars. And they coat this thing and they sort of make this part of the coronavirus, which is the part that triggers the infection. They make it somewhat immune to invisible to the immune system. Not completely so. And there is that one thing sticking out at the very top, which is called the receptor binding domain. And that is the part that interacts with what's called the ACE2 receptor in humans. And it's a key part of what makes this virus infectious. And I'll talk through that mechanism in just a minute. When the Pfizer and Moderna people devised their vaccines, they basically ignored this entire virus except for that region, that receptor binding domain. And they took a guess that that was the part to vaccinate against based upon, 20 years or so of research on coronaviruses. And they guessed right. Traditionally, what you would have done is taken this entire virus and turned it into just fragments, without maybe the ability to replicate, and taken all those, that neutered virus and exposed it to the human immune system. Instead, they just exposed this one little piece up here in these mRNA vaccines. And that tends to really focus the immune system on what's important. So if you've just been vaccinated and never been infected, you're likely to have antibodies against this guy. If you've been infected and vaccinated, you're likely to have antibodies against everything you see in this movie, okay? And so when a physician may, like, for example, we're doing a study right now on long COVID, we want to know who's been infected and who's not. We want to know who's been infected. We'll look for antibodies against other parts of the virus. We want to know who's been vaccinated. We'll look just for the parts of this receptor binding domain. Okay, so how does it work? Well, when the coronavirus encounters a cell that it can infect, and it can't infect all cells. In fact, that was one of the things that was worked out in the first several months of the pandemic, is exactly how does it get into cells. It attacks cells that have this ACE2 receptor. And so this is, again, the spike protein coming in, that receptor binding domain, which is now not really obvious, but it's this thing I've circled in red here, binds to ACE2. This cousin molecule, which also needs to be on these cells in this case, called TMPRSS2, comes in and actually cuts a piece of this spike protein, and it exposes a bunch of amino acids that really hate to be in water. And so what that does is it forces them to just stick into the cell membrane, because the cell membrane is composed of lipids that also, that's what the cell, it's hydrophobic as well, meaning that it also doesn't like to be in water. And so it sticks itself into that host membrane. And then this movie shows you sort of then what happens is that the virus basically will just merge with the cell membrane, penetrate in, put its infectious materials into the cell, and then the cell becomes a little virus factory, okay? So there's a lot of things that have to happen here. You need to have an ACE2 expressing cells. You need to have a cell that also expresses TMPRSS2. It turns out your nose, your bronchus, your lungs, and your gut all have at least some cells that do this. And then, and if you have all those conditions correct, then you can get infected, okay? So now, how is Omicron different from Delta, and what do those differences mean? So, while many people were taking a Christmas break, there was a lot of scientists around the world trying to figure out exactly the answer to this question. And the first thing that was done was to sequence, and we knew this from when it first popped up in South Africa. That was almost surely not the origin of this virus, but that was the first place it was detected. What I'm showing here on the left here This is the Delta variant of spike, just the spike protein. And up here is this receptor binding domain again. And I think it could be that this is that's the receptor binding domain. Okay, so these red things here say that one amino acid at this position 478 was substituted by a different amino acid. And the T and K and L and R just stand for different amino acids. And so this is how this spike protein differed from the initial Wuhan strain. If you look at the Omicron, it is crazy different, okay? And you can see all of these mutations up here. And what that means is that those patterns of sugars that were on the protein, on the virus, are going to change a little bit. The details of how that spike and the receptor binding domain interact with ACE2 is all going to vary. And the way in which that TMPRSS2 molecule comes in and cuts the thing is also going to be affected. So for example, if you have antibodies against the receptor binding domain of Delta, let's say you were infected by Delta, they're not going to act as well as they do against Delta, against Omicron, because Omicron simply looks different. It has different chemical composition at that place where the antibodies are going to react. And it's why also, if you'll remember, last fall and even throughout much of last year, monoclonal antibody therapy became a thing that was used in the hospitals for a lot of infected patients. And in fact, one of my colleagues who has an immunocompromised system was treated with monoclonal antibodies, and they worked wonderfully. But they don't necessarily work here because those monoclonal antibodies were developed against earlier strains of the virus. The new one is different. Omicron's different. Okay, so the good news is that the spike protein appears to interact more weakly with this cousin molecule TMPRSS2, the one that actually cleaves the spike protein a little bit, exposes those hydrophobic residues and forces, and then the virus just pokes into the cell, okay? So that's the good news. It also turns out that Omicron apparently does not need TMPRSS2 to enter cells. And what that makes it is a very good agent for infecting cells in your upper respiratory tract that do not have this molecule TMPRSS2. In the lungs, it turns out that the really deadly aspect of the virus was that it would affect cells in the lungs, and then you would get this structure here, which cells that get infected would actually become like one big cell with multiple nuclei. That's bad. That would cause a lot of cell death. It was a real, I would say a real pathology of the disease. Omicron doesn't seem to be able to do this very effectively. And so as a consequence, it can enter cells without the TMPRSS2. And so it has the ability to infect and generate virus in your nose and upper respiratory tract, but it doesn't seem to cause the type of damage in the lungs that the previous variants did. Now, I will say that, and you can, you all know this is true, that the Omicron variant's been around in the US for maybe, a month and a half. And so how could we know this much about it already? Well, this is, most of the data and results I'm giving you are from unrefereed scientific literature. They're from good groups, and so I trust the results they're getting, but one needs to beware. And so, the difference between unrefereed data and refereed data is that a referee will carefully look at a paper and may say, well, but you didn't do these controls. And maybe the group, no matter how good they were, just rushing to get what they think is important results out, didn't do those controls. So the story can change. But this is what the story looks like now. And it seems to have been replicated in a couple of these different pre-refereed publications. So it's sort of somewhat robust. Okay, so here's what happens in the air sacs in the lung and the bronchus. So here's Delta variant, and here's the Omicron variant. And so this is not a person. This is a hamster. So once again, buyer beware. But this is over the past couple of years, people have developed, they think, are really good models of coronavirus infection. This is one of those models, okay? And so if you look at the air sacs in the lung, after 3 days after infection, there's a little bit. 6 days after infection, you see it all over the place. You look in Omicron, and maybe there's a really little bit. So it really doesn't seem to be doing much damage in the lung. It's basically a factor of 10 difference. That's very likely will translate into the same thing that's going to be found in humans. In the bronchus, you'll see that there's a lot of virus coating here. And after day 6, you'll see that some of it's left. And basically, the Omicron looks maybe a little bit worse, if not the same. So that just backs up that statement I made before, that this can be more infectious but less deadly. Now, that's not going to be true for all people, and I'll put some caveats at the end. But it's largely going to be true, especially if you've been vaccinated. And so, a question you might ask is, well, I was vaccinated, and the Moderna and Pfizer and all the other vaccines that came out, but those are the two you want. They were initially designed against the initial Wuhan strain. How are they going to work against the Omicron variant? And it turns out they actually work okay. They don't work as well as if we were able to turn the clock back and expose vaccinated people to the Wuhan strain, which you wouldn't want to do that anyway, but they don't work as well. But here, what I've shown is the value of boosting as well as the value of having a vaccine. So here would be the amount of antibodies made, and I've tried to draw this red line here so you can see the comparison. So if you had a vaccine here, here's the vaccine here, and you were exposed to the Wuhan strain, you would develop in this NT50 unit. It's a quantitative unit. Don't worry about what it means other than that 10 to the 6th is 10 times better response than 10 to the 5th. But you would develop a pretty good response. If you look at the Omicron, you can see the vaccine actually does a lot. Your response is still almost 100-fold less than it would be if you'd been hit by the Wuhan strain. But in most people, that is enough. That's why people can get infected if they've been vaccinated, especially if they've been boosted. But it's why it is not that deadly. And I'll show you some numbers in a minute on that. And then here's data on mRNA and a booster. And once again, it's a similar story. Booster's great. Vaccine's great, okay? It actually gives you quite a bit of defense against this virus. And, I think in this country, we had maybe 65% of the people had 2 doses, at least maybe a booster as well. And people that were at really high risk, 80 or 70 years old, had the vaccination rate was much higher. And that's completely good news. So when you look at data like this, this is the kind of thing that you see on the CDC website, or you can pull it off the New York Times every day, or the Guardian, or whatever newspaper you read. These numbers that you look at they're both staggering, but they're also very much different from what we saw a year and a half ago. So here I've got almost 1 million cases a day. That's the staggering part. I've got 154,000 people, and I'll talk about that in just a minute, in the hospital. That's a big number. I've only got 2,000 people dying. That's a lot of people, but if this was a year ago, that number would have been more like 20,000. It was at 2% before, now it's at 0.2%, okay? It's pretty hard to interpret this plot because of all these cases, we don't know which one of these patients already had COVID, who was vaccinated, who was boosted. We also don't know all the numbers that are coming out of at-home tests. So the number of cases is almost surely higher, but the number of mild cases is also higher. But the number of deaths, it'll probably continue to creep up over the next couple of weeks, but it is comfortingly low. But is that comfort coming from the fact that we've all been vaccinated before, at least many of us have been, or because Omicron is less deadly? That's why I add caution about the fact that some of these papers were unrefereed, et cetera. They look to be true, but it looks like it is simply is a less deadly virus. So here's just some numbers for you. So this is put out by our own State Department of Health about 6 days ago. If you're unvaccinated, you've got about 3 times more likely to get COVID, 8 times more likely to be hospitalized. If you're less than 34, if you're between 35 and 64, I'd say 12 to 34. At 35 to 64, the odds go in your favor if you're vaccinated. And if you're 65 years old or more, and you should, your risk of death is 15 times reduced if you're fully vaccinated, and it's going to be better if you're fully vaccinated and boosted. And so what that's telling you, and I can tell you this is true, is that even though there are many, many people in the hospitals, in fact, our medical system is crazy strained right now, it's mostly filled with unvaccinated patients. And so vaccinated people, by and large, are getting past this Omicron variant without too many scrapes. That's not true for people that are immunocompromised or on certain preconditions, but as by and large, it's true. And so I put 2 jokes on here because I think we all could use a laugh. And I want to caution, oh gosh, my picture is hiding one of these jokes. This is an important one. I want to caution you that, I've given you basically epidemiological work and results and some, cell biology results. It doesn't mean that applies to you. It likely does, but it doesn't for sure mean. So for your individual health, you should consult your physician. Everybody is different. That's the point of the joke on top. Different immune system capacities, potential autoimmune situations and pre-existing conditions can make a big difference here. But by and large, I think the story I've provided you with is pretty robust. And I'm happy to answer any questions.
Great. Okay. Thanks, Jim. And just as, let me get out of this, stop the share. You can go ahead and start using that Q&A button at the bottom of your screen to pose questions to Jim. I already have one teed up that I'll ask of you, and that is about where do we move forward from here, and how do we protect ourselves in a pandemic but keep our lives going, the economy going? Where does this end? I mean, 2 years ago we thought we were going to be at home and shut down for a couple weeks, and here we are, and we're still dealing with this.
Yeah, and I think on top of that, the guidance gets from, for example, the CDC seems to change on a daily basis. It's really hard to know. I think the one message that is very strong is that if you are vaccinated and you are likely not to have serious illness from Omicron. And if you're vaccinated and boosted, it's even more help. And I believe that what's going to happen, for example, right now, the amount of people that are infected with Omicron is huge. We're running a study, part of a nationwide study on long COVID, and part of the study is to recruit patients at the very beginning of disease, and to also recruit never infected controls. And I think of the first 10 controls that were recruited into the study, 7 of them were not controls. They'd already had COVID. They didn't know. And so I think what's happening, and they probably had Omicron, or they were just, one of the asymptomatic folks and just didn't know about it in early variants. It's very likely we're all going to get Omicron or something like that. And it's also very likely that it's just going to become part of the, it's going to be become flu. And I think this Omicron is a first step toward doing that. It's still a very serious virus for many people. But I kind of look at this as a encouraging step toward returning to full normality.
Okay.
And I would expect that we would return to full normality by, I think within a few months.
Wow, you heard it here.
Well,
That's great news.
I'm a scientist, and if I wasn't optimistic, I wouldn't be a scientist. I always think the next result is going to be the best one. But I think the tendency to be more infectious and less virulent is probably going to continue. Doesn't mean it's going to continue linearly. It could jump back and forth, but that's where it's headed. And I think that the fact that we have now such broad exposure to both viruses, especially the Omicron, but also to vaccines, means that we have built in a level of immunity in the population, not in everybody. And if you're not vaccinated, for Christ's sake, go get vaccinated. But we built in a level of immunity that makes this something that you can live with and go about your life.
Great.
Okay.
You know what? I'll let you work through the questions, then I'll come back and wrap it up at the end. How about that?
So what is causing people with Omicron to die if the infection isn't getting into the lungs the same way? That is a great question. I don't think we know. I do know that, when we did just finish this study of long COVID, and we looked at 300 or so patients that had had COVID, and a fraction of them, and it's not zero, a fraction of them simply do not develop good antibodies. And if you look in their electronic health records, you'll find that they have some pre-existing condition that really is challenging their ability to develop antibodies. They may get over COVID because they have other immune weaponry, such as T cells, that can be effective. So my guess is that there's people that are just susceptible to the virus. Maybe they have, if you have a whole lot of temporary SS2 in your lung, you may not need to have such a great interaction with the virus to have that previous mechanism available. Most people probably don't have that much, but if you're an outlier on that particular protein, that may be the case. I think we don't know, but that would be a couple of hypotheses. That it's either immunocompromised people that are largely dying, or people that are really outliers in terms of the biochemical composition of certain cells in their lung. What is the short version of long COVID findings? The short version, and I think is really interesting, is that by and large, we can anticipate when someone is diagnosed with their initial COVID infection, if they're going to develop long COVID. And that's because there are several measurements we can do in the blood at the time of infection. Basically, they're almost, some of them don't pre-exist the infection, but some of them do, that are really the risk factors for developing, whether it's memory problems or fatigue or any one of these many, many long COVID symptoms. And those risk factors are, number one, autoantibodies. So we've talked a lot about antibodies in this presentation. Autoantibodies look exactly the same, but they bind to your own proteins. They don't bind to foreign proteins. And they will block certain immune response mechanisms in many cases, or they'll block some other biological pathway. So autoantibodies, patients that have those and I think lupus and there's other autoimmune diseases where they're very prominent, very likely to develop long COVID. Most people that have autoantibodies don't know it because it's what's called subclinical, means that you've just been living with some, you may not have a lot of them. They haven't really manifested themselves in a way that would have encouraged a physician to measure them. Another one is the actual genetic material from the SARS-CoV-2 virus that's in the blood, not the nose, but in the blood. And that probably is a sign of infection severity and just escape of the virus throughout the body. A third indicator is there's another virus that almost all of us have, 90% of us have this. It's called Epstein-Barr virus, well known as the one that triggers, that causes mononucleosis, the kissing disease that puts you on your back for 2 months if you're a teenager. That virus can get reactivated by the SARS-CoV-2 infection. And you can see that virus genetic material also in the blood right at diagnosis. It goes away quickly, but you can see it right then. And then the third factor we measure is the pre-existence of type 2 diabetes. There's probably other ones. We didn't do a comprehensive survey, but we did it was pretty deep. And each one of those accounts for a certain fraction of patients that get long COVID. Autoantibodies count for the most. So that's the short version. Okay.
You have 16 more questions.
Any evidence that COVID-19 integrates with the host genome? No, I don't think so. I don't think so. Yeah, I don't think so. Do we have an idea of how many more cases there are than are officially reported? Sorry, I missed if y'all were talking about it before. I don't think anyone really has an idea. I would loosely guess a factor of 2 or something. And if you think about that, that means that the number of people reporting COVID per day is roughly the number of people who are getting vaccinated per day during the peak of our vaccine period. So that's a big chunk of the country. What is the nature of the sugars on the spike protein? And how are they used by the virus? So the sugars are what's called glycans, and they are, they're not like sucrose or the kind of sugar you would get in your, but they're similar. They're long carbon chains with a lot of OH groups on them. And OH, if you think about structure of water, H2O, that has OH 2 times. So that basically makes the virus water dispersible. Basically gives it a hydrophilic nature so that it disperses inside water. But it also masks the virus from the immune system. So that's basically the chemical formulas. They look like long lipids with just lots and lots of OH groups on them. And they're just used for solubility and promote immune evasion. What are you seeing with long COVID and Omicron versus Delta? That's a great question. I think it's too early to know. I almost can tell you for sure it's going to be different. And probably will be a little less severe, maybe even more than a little. If you look, there's some people have done epidemiological studies of long COVID, meaning just population-based health measures, from the Wuhan strain through the Alpha, Delta, et cetera. And they've seen evidence that each of these strains of coronavirus causes slightly different long COVID. Now, that's what I call buyer beware research, because you don't know if they've corrected for rural versus urban or ethnic, or, there's all sorts of corrections that one has to account for to understand the comorbidities and other things that were present but it looks like to some level, long COVID does depend upon the genetic nature of the virus. And we also know that it depends at least a little bit on severity of disease. And so with Omicron causing by and large less severe disease, one would expect that Omicron is going to have less long COVID associated with it. Okay, but we don't know. It's too early to tell. Thanks for the nice talk. I have a question about how the current vaccines, Moderna and Pfizer, et cetera, could work with the new Omicron variant, which has a big difference in the structure of the spike protein. Well, they make the antibodies that are made in response to these vaccines do still see the spike. They just don't see it as well. So it's not like a black and white. Either the antibodies work or they don't work. If they don't work as well, you can overcome that with more antibodies. That's why boosting is important, because basically you're just trying to, you've got a spike protein here. The antibody, you want to come in and block and prevent that spike from infecting a cell, and then it also tags the virus to be cleared by the immune system. And if it doesn't bind as strongly, it'll come off. But if you got a lot of antibodies, one's going to be around to come on again. And so the more antibodies you have, even if they don't work as well, you'll still get an effect. And that was why that boosting is kind of important. I would have said for Delta, probably boosting wasn't so important, but I think it actually is for Omicron. What do we know about the etiology of long COVID? Do you see any similarities with ME/CFS? I think that the question you're asking, Jill, is there are other diseases that have long effects. Famous ones are patients that have had serious sepsis. There's chemo brain that can come from chemotherapy and radiation therapy. Post-acute Lyme syndrome is a very famous version of this. Before the pandemic, we were working on launching a study on Lyme to try to understand post-acute Lyme syndrome. I would completely redesign that study based on what we learned about long COVID. And in fact, I would really focus on autoantibodies. I think that they actually are going to be very important. And I would look at other signatures as well, like I talked about, maybe other comorbidities. But I'm guessing that that a lot of these long diseases are really related to perhaps subclinical autoimmune effects that people maybe didn't know about, but actually have an impact on how they progress through the disease. Okay. Do you anticipate more variants like Omicron that are so vastly different from earlier forms of the virus? How likely is a variant that can sidestep our vaccines and boosters? There will almost surely be more variants. I mean, it's like we were, I guess, caught off guard by Delta. We shouldn't have been. And then it's like, holy smokes, there's another one, Omicron. And the chance of getting a variant is directly proportional to the number of people that are infected, because that's how the virus mutates and the mutations can spread. And so almost surely we will have more variants. It is at some point for sure, the vaccines that we got based on the Wuhan strain are not going to be functional. They're still functional now, but they won't be functional at some point in the future. But Omicron's so widespread that may be a surrogate for vaccination. And there are vaccines being developed on some of these more advanced strains right now that I think will be provided as boosters in the future. So I think this is something we will just learn to live with. So I think variants will eventually sidestep our vaccines, But our vaccines will also keep pace, our old vaccines, but our newer vaccines will keep pace. And these mRNA vaccines are, really impressive technologies. And that was really, I think, accelerated by the pandemic. So it's a silver lining. Thank you for the excellent presentation. You're welcome, Bridget. I'd be interested to hear if you think that the efforts to educate Americans about COVID vaccine safety are helping those on the vaccine hesitancy continuum with other vaccine preventable diseases. Well, I'm not a sociologist. I'm still stunned to find that there are people that are not vaccinated, especially after what this country is going through now, and just looking at who's in the hospital and who's not in the hospital. I'm hoping that that sort of education by fire will have a profound impact for things like coronavirus. I can understand people's hesitancy against getting vaccinated for everything that comes along, because people don't like to put foreign things in their body and things like this. But this is a case where the science has been it's like polio. It's been such a clear example of success. And then the dangers of not being vaccinated are so clear now as well, that I'm hoping that this education by experience will pay off. I'm not that optimistic. I'm hoping. How are neutralizing antibody levels related to long COVID? Can asymptomatic infected get serious long COVID? Thank you. Oh, there's Drew. Hi, Drew. Yes, asymptomatic infected patients can get long COVID. We found that many of the long COVID symptoms that were, say, associated with autoantibodies, they were still associated with autoantibodies when you corrected for disease severity. And it's not just a taste and smell. Those actually you can have loss of taste and smell without having serious respiratory disease. But some of the other, like fatigue and inability to exercise, don't seem, and even some of the brain fog, don't seem to really correlate necessarily with disease severity. There is a disease severity role here, but you can be asymptomatic or low symptomatic and still have long COVID. I don't know if we have enough data to know how serious long COVID you would get, but it may be something that would clear up after a few months. I don't think that's clear yet. As we are using antibodies, but viruses mutate themselves to protect, why should we not target its replication complex by drugs to inhibit that action? That's a good question. And I would say that, some of these antivirals that were just announced by both Pfizer and Merck actually kind of do that. And in fact, and I mentioned that even long COVID depends upon these very early loads of both SARS-CoV-2 and Epstein-Barr virus in the blood. One might expect that antivirals used very early in the disease might have an impact. But the trick is you have to use them really early. And many people, by the time they start feeling sick enough to go see a physician, get tested, you're too late for the antivirals to have an impact. So I think it's a great question, but I don't know. I think it's a challenging one to treat patients in a timely fashion. And Annie asks, have you heard if there are other variants waiting in the wings from elsewhere in the world? I've heard of a couple variants on Omicron, but I haven't seen, what you would call rock hard scientific data that suggests that these are going to be scary variants as well. So I would say just, wait, tick-tock, tick-tock, tick-tock. But like I said, I think the next one is likely to be less virulent or less dangerous than this one. At least that'll be the trend that we see. Anonymous, among the people with COVID, are there any groups that do not seem to have a risk of developing long COVID? Well, if you don't have any of those three things that one can measure early on, your chance of developing long COVID is much lower. It's not a great diagnostic, but if your viral levels are kept at a low level, so let's say that's for one of those virus levels, that's going to be if you had mild disease. If you don't have autoantibodies, most important thing, don't have autoantibodies, and don't have type 2 diabetes, then your chance of developing long COVID is going to be greatly reduced. Perhaps you mentioned it, but what is the mutation rate of the virus? I think the mutation rate is likely proportional to the numbers of people that are infected. And so one would expect this virus, Omicron virus, has a mutation rate that is significantly higher than the Delta variant. It doesn't mean that we're gonna see a new variant emerge faster, but it kind of suggests that might happen, and I wouldn't be surprised. What have you? I guess coming back to David's question, the mutation rate. If you're infected, I'm infected. 20 people are infected, the mutation rate of the virus in all those 20 infected people is not the same. And there are individuals, and this is something that was found out during the pandemic, that seem to be sort of super mutators. Their body really supports crazy amounts of mutations. And so it's the virus finding those individuals that are, obviously proportional to the population, but they're relatively rare individuals that have this type of sort of super mutation capacity. And they're immunocompromised people where the virus just keeps replicating, and maybe there's some oxidative stress going on and that promotes mutations and things like that. I heard that COVID can get into almost all the tissues of the body. How does that work? I would say you can probably find fragments of the virus in all the tissues of the body. It's not obvious that you can get active virus in but just a handful of tissues, like the gut, the lungs, and the bronchus, maybe a couple of other places. People looked pretty hard for that and haven't seen evidence of it. So now it could be that people with long COVID, and especially people that have, unusual symptoms, that they may find evidence of competent virus in some tissue that we don't know about. But it's competent virus that one worries about. So just seeing a signature of COVID, for example, when we see the virus in the blood, by and large, you can take that blood and expose it to cells, and they may make those cells may actually sort of kind of turn on the machinery. They'll make one or two proteins of the virus, but they won't make virus. So it's not competent virus. And so if you thought about, well, gosh, if people have virus in the blood, that sounds like it's a pretty great spreading mechanism, or virus in the gut. Probably not. Probably not. It's probably not competent. Okay, is there any data that an individual can get reinfected with Omicron or Delta? Almost surely one can get reinfected with Omicron or Delta, depending on the level of antibodies that you make. I think let's talk about the difference between having had the virus and having had the vaccine. So when you have the virus, your immune system will respond And as I mentioned earlier in my talk, you'll develop antibodies to all aspects of the virus, the important ones and the unimportant ones. Now, your immune system has a certain capacity. And so what you've effectively done is you spread that capacity over 100 different little regions of molecules in the virus where you can develop antibodies. When you have the vaccine, you are focusing your immune response on just that receptor binding domain of the spike protein. That's good and bad. It's good because the antibodies you have will block that. It's bad because if the spike gets mutated so it's no longer recognized by those antibodies, then your body that does nothing that recognizes the virus really. By and large, I would say that that's been good. In fact, I would say not just by and large, it's been very clearly good, that limitation. But it is possible to get reinfected if you had a weak antibody response against your initial infection. But you can boost that up through vaccines. There was a sort of a thought in immunology for many years that there's no better immunity than actually getting infected with the virus. And I think these vaccines have actually challenged that concept a little bit. Okay. So Shannon asked, there is approximately 95% less chance of complications and death if one does not have any comorbidity factors. Bolstering that percentage would be a strong immune system. The immune system, can you scroll up a little bit? Oh, I can scroll up a little bit.
What's the right? Or is it right?
It's the last question here.
Oh, there we go.
Okay. The immune system has been shown to be negatively affected by the vaccine, and thousands of people have had complications just from taking the vaccine, though that is not publicized. Okay, let me read this again. There is approximately 90% less chance of complications death if one does not have any comorbidity factors. Bolstering that percentage would be a strong immune system. The immune system has been shown to be negatively affected by the vaccine, and thousands of people have had complications just from taking the vaccine, though that is not publicized. There have been complications with the vaccine for sure, and you can imagine, people that get knocked flat on their back for a day, people that don't get anything, and then there's people that might get more seriously impacted. I think you could come down decisively that there have been some complications with the vaccine, and you're right, Shannon. The number probably is in thousands, but the number of people that have been vaccinated is in the many, hundreds of millions. And so statistically, I think you can make a very strong case that having the vaccine is a major bonus. There are patients who have compromised immune systems to begin with, and there's certain things that those patients need to worry about, whether they're getting vaccinated or being exposed to the virus. And so I would guess that just like some of the comorbidities that lead to serious disease, there may be comorbidities that aren't really well known yet that lead to a side effect from the vaccine. It's rare enough that it makes me think that probably it is it's something that one could detect ahead of time, but I don't know if there's been enough patients with such adverse effects that have participated in studies to know what those effects might be. But I think your point is a good one, but I'm going to strongly come down on the fact that I think that the vaccines are still the best option.
Is that our last question?
That's the last question.
Wow, good job. You did, look at that. We did this right on time.
4 minutes to go.
Nice,
Cool.
nice, good job. So thank you, Jim.
You're welcome.
And we would love to see everybody. We have a couple of events coming up, and the next one you're going to be at Town Hall Seattle.
I am.
Yeah.
I don't know, if you can see, but I'm talking to, gotta
There
hold
you
it
go.
so I can
Yep.
see it. Matt Rechtel, he's a Pulitzer Prize winning journalist who is a really interesting guy. So he wrote about the immune system, which is how I encountered him. But he's also wrote about some really interesting things. Like, and I think people who do Zoom calls all the time can appreciate this. One of his books was about texting and driving and the laws that led to don't text and drive. And the issue was, can you pay attention to two things at the same time? And what is the psychology of that? What is the sociology of that? What is the biology of that? And so we asked that question in quite a bit of depth within the context of some dramatic story. He's got this book on the immune system, but he's also got a series of articles now coming out on in the New York Times, I think just starting at the end of this month, on the impact of social isolation on some of, on basically on our younger generation. That has been a serious and detrimental byproduct of the pandemic. So I think it'll be a wide-ranging discussion. There will be no slides. We'll just have a fireside chat.
Great. That's Thursday, February 10th, and that's Town Hall Seattle, and it's a virtual event, so you can watch it from the comfort of your home. You will need to get a ticket through Town Hall Seattle, and they're $5 each. And then our next event, and last one I'll talk about, Thursday, February 17th is our next research roundtable that will be featuring ISB's Dr. Nitin Baliga, and his topic will be finding new drugs for ongoing tuberculosis pandemic. So thank you, everybody. That's all we have for tonight. Thanks, Jim.
Thank you.
Yeah.